And it's my pleasure to introduce her. Actually, it was quite funny how we got in touch. I saw one of her talks on YouTube, and I was really interested in it. It was about insect intelligence. And then I thought, I'll just drop her an email and see if she answers and if we can get in contact. And it worked, apparently. So now we have a project together, and we are working together, and she's one of the distinguished experts in intelligence in insects, and especially ants. So I'm really looking forward to the talk. Thank you for being here, and yeah, have a nice talk.
Okay, thank you, everyone, and thanks very much. Thanks very much for the invitation to speak to you today about my work. And perhaps a little differently to what we just saw introduced, which was a lot of really interesting and often bio-inspired work. My kind of approach is really, the reason I build robots is because I want to understand the animals and how they're doing what they're doing. So what's actually going on in their brains when they do behaviors such as the ones I'm going to talk about. Navigation, so this is a desert ant going home. It's not using a chemical trail. It's just finding its own way through visual memory and path integration. So I'll talk about work we've done on that for quite a few years now. And then I'm also going to talk at the end about one of our new projects, which is on how ants grasp objects. So my inspiration for this work, and I work on other insects as well as ants, although I'm going to focus on ants today, is that insects do a lot with a very tiny brain. Their brain is maybe 1 millimeter cubed. If you're familiar with fMRI, their entire brain would fit within 1 voxel of fMRI. So we know it's on or off. Okay? They have about 100,000 neurons, depends a little bit on the species, but it's a very structured brain. It's not just a whole mass of stuff. There's a lot of structure in there. And interestingly, many of the circuits in their brain are the same across different insect species. And if you think about the variety of body plans, of motor outputs, of lifestyles of insects, it's a huge, they fly, they crawl, they swim. Yet many of these circuits seem to be the same, which suggests that maybe they're quite useful circuits for controlling behavior. So we might learn something for robotics by understanding their capabilities. So as I said, the work we've been doing previously, our focus has really been on the navigation behavior. And so this is a desert ant again. So these ants forage individually because it's so hot in the desert, they can't lay chemical trails, they just burn off, and also they have to move very fast. And this is wonderful tracking behavior that one of my postdocs did of this ant going through this quite complicated terrain, you can see, and they can go hundreds of meters away from their nest through this terrain, and their nest is a tiny hole in the ground, and they find their way back home again. So it's really quite an impressive navigation, individual ant navigation capability. This is just from these tracking studies. This is a whole set of ants, and you can see each of them is going out in a different direction. And in this tracking, we're actually able to reconstruct both the track of the ant. We're following the ant with a handheld camera, and the ground underneath it. So we can start to actually get into the details of, what do their paths look like, and how do they go around bushes, and what do they encounter. We can get right down to really fine scale information about what the ants are doing. And one of the key things that they do is, as I said, go to and from their nest. So this is, in this study, we were actually following ants from the very first time they ever came out of their nest. So we knew that the ant was naive, it knew nothing about its surroundings. And what it does when it first goes out, I'll try and use the pointer here because I know people online won't see if I point over there. So let's hope this works. So the first time they go out, they'll be maybe doing a complicated route. They're just kind of foraging, wandering around looking for some food, which is fairly sparse in this environment. And in this case, they found food at number 1 here. So then what they do, when they get the food is go straight back home. So as you saw before, there's bushes and everything else around here. So they're not taking the outward path, they're going straight back, in the direction that they were going, where their food is. And they know when they get back to the nest, they don't overshoot, they'll start to search once they get to the nest. And this is called path integration. You might think of it as odometry or dead reckoning. They're basically keeping track of their velocity for the entire path, integrating that velocity so that they continuously have a vector pointing back home. And I'll explain a little bit later in the talk how they're doing that. And also, once they have got the food and got back to their nest, they don't come out and wander randomly again. Instead, they then go straight back to where the food is and come back home again. And if you then are a bit mean to the ant, let it get home, and you just pick it up at the nest and take it back to where the food was. So now it doesn't have the path integration. Its path integration tells it it's at the nest, but it's actually at the food. What we find, we release it, is that it does a quick search, and then it follows the route that it's followed the last 2 times back home. So even after just 1 or 2 times going along this route, it's actually learnt this route visually, and it can follow it home again using visual memory. So we can maybe compare, what these ants are capable of to self-driving cars, which require conventionally structured roads. They get a detailed 3D map from large-scale advanced data gathering, whereas, as I've just shown you, these ants can deal with entirely novel, unstructured terrain, uneven ground, vegetation. They have minimal prior knowledge. And also, they have a surprisingly different sensor systems. So self-driving cars typically have multiple high-resolution cameras. They have range sensors, laser, radar. They have accurate internal motion measurements. They have odometry. And in fact, really critical for self-driving cars to work at all has actually been GPS as well, and using wireless information as well to locate themselves. Whereas insects appear to have quite low-resolution vision. It is omnidirectional, and it has a few special properties that we don't have in normal cameras. One is that they're sensitive to ultraviolet light, and another is that they're sensitive to polarized light. I'll just talk a little bit about the polarized light at the moment because I'm going to skim over it a little bit later, but it's actually quite important. So the insect in the top part of its eye has a little area where the receptors actually have aligned microvilli, which makes them sensitive to the polarization direction of the light. And they're pointing at the sky, and the sky is polarized relative to the sun position. There's rings of polarization direction. So you can actually use this information as a compass. And a lot of insects do actually use this as an external compass sense, which is very important if they're trying to maintain an estimate of their velocity, so their direction. Okay, but I'm also now going to go just back to ultraviolet. Why would ultraviolet light be useful? And this was just a little experiment we did, really, which was to take a conventional camera, take out some filters, put in some other filters, so that we could see what happens when you have pictures in ultraviolet. And this is in the ant environment, and this is in Edinburgh. And hopefully what you can see here is that what's really distinct between those two, the ultraviolet and the non-ultraviolet, is how much ultraviolet there is in the sky. So the sky has a lot of ultraviolet light, and the ground has, in general, very little. And what we found is that if you were trying to separate sky from ground, and you do it in UV space, it's like night and day. It's really, really easy to tell what's sky and what's not sky. And actually, over even in cloudy conditions, the UV comes through the cloud. Even in quite late evening, when it's still not a lot of sunlight, there's still this very sharp boundary. Whereas if you try and do this in RGB space, you can see there are clusters. You can kind of separate sky from ground in RGB space, but it's quite a lot harder to do that separation. So this is just one example of how we use this. So on this camera, we have a couple of GoPros, one of which has UV filter, and the other one doesn't. And they got driven around these quite long areas with quite uneven terrain. So this is looking omnidirectional camera looking up, and you can see the RGB and the ultraviolet. And then we just do this simple filtering. We literally just set a threshold, and we turn this into simple binary images. And then the idea is each of these binary images tells you something about where you are. So the pattern you can see in that canopy is actually surprisingly recognizable. So we use the method called sequence slam, where you compare a sequence of images to try and get a good match. And we found that if we did this UV sky segmenting, it worked really very well to locate yourself over these long trajectories. So that's one reason to use UV. If you can take essentially the horizon pattern is a very recognizable pattern that you can use to locate yourself. So if you're an ant and you're trying to follow a route, how can you do this? And one of the key ideas that was actually suggested by some colleagues of ours at Sussex University is that the insects are not necessarily recognizing where they are. What they're doing is recognizing that things look familiar. So if you imagine, as you do, when you get to a new city that you've never been to, like Esslingen for me, one way to not get lost is to, when you're kind of leaving the station or whatever, if you look around and look at what the world looks like as you leave, then when you're coming back, you'll get this recognition sense, and you'll know, oh yeah, I'm going the right way because things look familiar. Okay, so that's in essence the algorithm that was suggested here, is that you store every view when you first go along the route. And then on the next one, at each position along the route, you basically can scan back and forth and say, when do things match? When do things look familiar? And that's the direction that I should go. So that's what I'm sort of trying to show here. So we have a set of images that we've collected along this route. And then at each point, we're scanning to the left and the right. And as you can imagine, if you're pointing in exactly the same direction, then you'll get a zero difference, so this is just the image difference. But you also get this nice valley that as you're slightly to the left or slightly to the right, it increases gradually the difference. So all you need to do then is essentially run along this valley, and you're following the route. So that's the concept. We've tried to use this on robots, and this is just a couple of little examples. So this was one of our cheap and cheerful robots. It's actually a mobile phone on wheels with a little omnidirectional lens on the camera. I think it's going to go to that in a moment, and you'll see. And it's got a very low resolution vision, but it's basically remembering what it saw all the way along this route. And you can see here we have this little mock-up of the ant environment to test it out. But we also tested this back in the real ant world. So the robot couldn't go over the uneven ground, so he put the boards down, but otherwise it's using this strategy to follow a route that it's learned, even though the wind is blowing stuff around and everything else. So that's nice, but how could that ant's tiny brain actually implement such an algorithm? We're storing hundreds of images here and then doing a comparison. For each time we see what we see now, we're comparing that with everything we stored. Is that a reasonable thing to be going on in the brain of an ant? So the basic idea is that the ant should associate the views it experiences with its progress towards the food or towards the nest. And there's a part of the insect brain that's now being studied in a lot of detail, which is actually studied for olfactory pattern learning. But in ants, this part of the brain actually has a lot of visual input as well. So it's an area called the mushroom bodies, because it looks a little bit like mushrooms up here. And as you can see, this is the eyes of the ant, and this is the visual processing, and a lot of information goes in. So our idea was that maybe that's where it's learning this visual information. So what we do is to test computational models of this brain area to see if it can actually support navigation tasks in the way we said. So I'm just going to talk through this fairly quickly, and I'm very happy if anyone wants to get into more detail later on. So it might look quite complicated, but actually conceptually it's a relatively simple idea. So as I said, this has been studied for olfactory neurons, and this is an area of the insect brain that we now have a complete connectome of. So what that means is we actually know every single neuron, every single synapse in this brain area for Drosophila, I should say, not for ants. But that gives us a lot of information to be going on with. And what we know is that the sensory inputs come through a relatively small number of projection neurons into a very large number of Kenyan cells. And in the fly, it's something like 50 and 2,000. There's something like 200,000 of these cells in the brain of the ant. So what this means is that we're taking a sensory pattern and we're projecting it up into a much higher dimension, but using sparse coding. So we have a sparse, high-dimensional encoding of the pattern. And the main reason for doing that is then that separates, every pattern we see gets separated, and we can learn each of them in a very simple way. So here, what we're doing, sorry, my mouse is coming and going. What we do is we learn which of these neurons are active, which of these few neurons are active for any one particular pattern, and we associate that either with going home or with going out from the nest. So we have two output neurons here. And then when we see the next image, we just change those synapses, and we see the next image, we change those synapses. So gradually we're increasing the number of synapses that represent all the patterns we've seen so far. So then when we see a new pattern, so here we have visual input. When I see a new pattern, if I've seen that pattern before, these synapses will have already changed and I'll already be getting a different output. So I know that I've seen the pattern before. So really, I'm just doing familiarity. I'm not recognizing, I'm just saying, is it one that I've already learned or is it one that I haven't learned? And it turns out that you can store hundreds of images in this circuit reasonably easily. There's something else I was going to say. Oh, yes, and just to say, although here we're focusing on images, this whole part of the brain is actually multimodal. So you could imagine that you don't just have to learn what the world looks like. You might also learn that when I'm in this place, there's a particular smell, and the wind is coming from this direction, and the ground surface feels like this. So all of those things kind of form a gestalt of this is somewhere familiar versus this is somewhere I've never been before, or this is somewhere that's vaguely familiar, so maybe I should keep going in the direction where the familiarity is increasing. And we've now updated this model. This is the most recent version of the model where we're actually also learning sequences. So we learn one of the things from this connectome where they've traced all these synapses. They found that these parallel fibers of these sparse representations seem to connect to each other. So we use those to actually try and learn when we see a sequence of images what that sequence is. And that improves the recognition. So this is a robot, again, moving through an outdoor environment. In this case, we're actually using an event-based camera to detect edges and moving edges. And we store those in this memory, as I said before, and we actually use neuromorphic hardware to run the algorithm. And we're able to detect, here we've learnt a small section of the path, and then the rest is unfamiliar. And we get a very different output that lets us know, are we on the familiar path or are we not? Okay, so that was a bit of a whirlwind introduction to one bit of the insect brain. I'm going to give you another bit of insect brain that we're also enjoyed working on recently, which is a part called the central complex, because it's in the center of the brain and it's complex. And this spaghetti that you see here is the connectivity that's there, and that's all being traced by my collaborator Stanley Hines at Lund University. And it does just look like spaghetti. How could we make any sense of this? But actually, there's a lot of structure in here, and you can maybe see a little bit even in this kind of, biological diagram, that there's actually these columns running through here, and we actually have a very regular pattern of connections that come from the columns and then go across and then go into these different layers in this central part of the central complex and so on. So we've actually spent a lot of time on this circuit, and in the end we think we've really understood what this circuit does, and we think what it does is the path integration that I showed you earlier. So I'm going to, as I said, again, give a kind of whirlwind tour. So this is our connectivity diagram, and this might look like, okay, now this is just a completely, little artificial simplified diagram. Actually, every connection in this circuit that we've built, we can justify it as a connection that's in the real brain of the insect. And what it has is a set of neurons that respond to the heading direction relative to this polarized light compass, which is the central circle. It has neurons that respond to optic flow, so that it gets the speed and the direction. And those sets of neurons actually coincide, their inputs coincide on this ring here of neurons that we call memory neurons, which are basically, we assume what they do is they integrate. So they integrate the speed relative to the direction. So you're getting a distributed representation of your path. And then finally, on the output side, we have these steering neurons. They get information from the memory and from the current heading, and by comparing those two can decide which way to steer. So let me just run the little video showing you the kind of simulated, in this case actually a simulated bee rather than an ant, but we think it's exactly the same in ant behavior. So you can maybe see that as it goes, the direction it's going is in the inner ring, and the further it goes away from the nest, the stronger the activation becomes on the side pointing back towards its nest. And then when we let it control its steering to go home, it takes a beeline back towards the nest. And also, because the memory is continuously updating, when it gets close to its nest, it will actually kind of zero its memory. If it overshoots, it will pull it back. So we get this search behavior emerging for free as well. So that's nice. Let me just say a little bit more about the speed encoding, because it's actually quite clever what happens. So how this got discovered, again, this was my colleague Stanley was recording from neurons in the bee and showing them effectively Star Wars videos of the world going past like this, or rotating around them. And he found specific neurons that responded very well to this front-to-back motion, as though the animal is flying forward. And they didn't respond to any other kind of motion. And moreover, they responded in this very linear way. The faster you were simulating flying forward, the stronger the response of these neurons. So that seemed like a good speed input. But what he found then, when he said, what do these neurons really respond best to? So what he did was to move a stripe around and say, when does the neuron respond best? Which direction of motion? And what he found is that for the neuron on the left, it preferred motion from 45 degrees going this way and from 45 degrees going that way. So its preferred motion was actually as though you were flying 45 degrees sideways, okay? And the same for the other neuron. It preferred flying this way. So you weren't getting best response to forward speed, but to flight in two different directions. So why have this offset in the preferred optic flow? Actually, this works very nicely if you're not going in exactly the direction that you're facing. Okay, so that's true for flying insect, but it's actually also true for ants. They're very often carrying things, moving around, dragging things, not going in the direction they're facing. So here's the kind of intuition. If the animal has a particular heading direction, so that's the direction in which its head is pointing relative to the sky, but it's actually flying or moving in a slightly different direction here, then because it has these two neurons that respond best at 45 degrees, you're basically projecting its actual motion vector onto an orthogonal basis. That means you can tell where you're going. So between the two sides, you're actually getting your real velocity information. So the left and the right sides of the brain can represent the holonomic motion of the animal relative to the ground, and combined with the compass sense from the polarized skylight, you're actually getting a geocentric reference frame of your motion, okay? So the insect actually knows where it's moving with a geocentric reference frame centered on its nest or starting position. Okay, so we wanted, of course, to test this on a robot. So in this case, we're testing it on a flying robot. We are actually working on reproducing this polarization compass sensor. You can ask me about that if you like, but for the moment, we're just using a magnetic compass. But we use a camera that's pointing down at the ground with a gimbal to keep it facing the ground. And we have a computer that's running this neural network that I showed you before. And we're trying to detect the optic flow to estimate the ground speed. And we do that, as I described before, by having this kind of sensitivity to 45 degrees. So we have what we call match filters that filter this optic flow for 45 degrees. And we need to compare that to a depth map, because obviously the speed you're going above the ground, how fast the optic flow is, depends on how high you are when you fly. And when we do that, and we try and extract the X and Y position relative to a kind of ground truth, then we find it actually works surprisingly well. So without the gimbal, it's very noisy, as you can see, although still the X and Y positions follow quite well, the actual X and Y positions. And once you have this stabilizing, then actually we get extremely good estimate of our position, X and Y movement, as we go across the ground. And so then we obviously test this for path integration. So this is the flying out with a kind of wobbly path with a few large turns in it, going hundreds of meters away, and then trying to return back using just this neural circuit that I showed you, which is less than 100 neurons. And it's able to find its way back. And obviously we do multiple tests. We measure the error. We can also simulate this to give different types of ground surface and so forth, but the key thing here is the blue line and the blue dots, which are from the real world tests, and what you can hopefully see is we have a couple of meters of error when we fly, 150 meters, which we think is pretty good response. And this is in windy conditions. We haven't over a field that has no landmarks or anything in it. The height compensation is quite important. It works well up to, even if you vary the height by 2 meters without height compensation, it still works pretty well. Beyond that, it doesn't work so well. And we have two theories about what insects might be doing here. So if it's an ant running above the ground, it's not a problem because its height is relatively constant. And in fact, bees tend to fly at a fairly constant height as well. But there's also some other ways in which we think they might be able to estimate their height. In the interest of time, I'll skip that for now. Okay, so that's navigation behavior. I'm now going to switch a little bit to something that's a current project and much less far advanced in terms of what we understand, but hopefully to give you the flavor of what we're interested in right now, which is skillful manipulation in ants. So my group actually does field work with the ants, where we're trying to do these navigation experiments where you pick up an ant and put it somewhere else and see what it does. And when you do that, you spend a lot of time watching ants, because you mark the ants and you're waiting for a particular ant to come back to the feeder. So I'm sitting there watching the ants picking up food from the feeder, and it occurs to me they're really very, very good at picking things up. And this is just some of the more advanced things they do. They also cut. So leaf cutter ants actually cut little bits of leaves and then pick those up and take them back to the nest. Ants, this species of ant, sorry, I'll use this pointer down here. This is one of its larvae, and it's squeezing that larva to extrude this substance that allows it to sew the edges of a leaf together to build a nest. So it's doing sewing, and it's not killing, the larva. It's very delicately holding this larva. Of course, ants will hold on to each other to cross bridges, or to, in this case again, to kind of close a leaf over. And then if you imagine, what goes on in the nest of an ant, just to build the nest, they have to do a huge amount of manipulation. And then again, they're doing a lot of things like looking after their brood, bringing food in, taking rubbish out, and so on. And this even extends to using tools. So this is one of my favorite recent studies, where they had ants and there was sugar water that the ants could pick up. But sugar water, an ant can only drink so much and take it back to the nest. So what they did is they gave the ants a choice of lots of different objects that they could drop in the sugar water, which would soak up some of the water, and then they could take the objects back to the nest. And the ants very quickly learned that these little sponges were much better than the little sticks, for example. So they could actually pick which of these tools was the most effective and then use it to carry things back. Okay, and roughly speaking, if you look at ants picking up food, what they're doing is executing a parallel jaw grasp of unfamiliar objects in clutter. So this is just one example. This is slow motion filming of what the ants are doing. Here's another example, even in more slow motion. Sorry, it's a little bit fuzzy, but this is just to kind of illustrate that they're doing this, in a cluttered environment. They're making a distinction between what they should pick up and what they shouldn't. Usually making quite a quick decision. And you can hopefully see their jaws that they're using to pick things up and carry them around. Oops. And picking parallel jaw grasp of objects in clutter is actually still a key problem in robotics research, as I'm sure many of you are aware. I quite like this recent quote from Jeff Bezos. If you went back in time 30 or 40 years and asked people what problem would be harder to solve, machine vision, natural language, understanding, or grasping, most people would have predicted that grasping would be solved first. But in fact, we've more or less solved machine vision and natural language understanding, but grasping, we're still way behind biological systems in our capabilities. So as I said, it's effectively a parallel jaw gripper. So this is the ants' grasping equipment. They have a single degree of freedom, hinge lever, mandibles. They're very strong. The muscles actually occupy nearly all of their head capsule. Their head is much more full of muscle than it is of brain. Basically, the brain is a tiny bit inside the head. They use their, but of course it's a 6 degree of freedom head and it's a mobile base. They have their antennae, but also lots of tactile hairs and obviously force sensors in their muscles and cuticle. It's unclear to what extent they're using vision in this. They do manipulation in the nest, so they don't require vision. They may be using their legs and antennae to some extent to position the object. But I'm pretty certain the brain is not doing the standard approach in robotics, that is, it's not doing 3D reconstruction of objects and grasp planning. So this is the kind of set of research questions we're pursuing at the moment. How hard is it? How many good enough grips are there? And can we give the ants difficult to grip items? Do they make good choices about where to grip? There's very little study of the biology of the system. One paper where they found that there were seeds that had little shoots off the seeds, and they would always pick it up by the handle, effectively, and move it that way. What precision of control of their force and grip are they capable of, and what precision of placing their grip? Do they do stereotype poses and movements? What sensory information do they use? And we're also very interested in whether they improve with experience with the same objects. Do they get more and more capable of picking them up? So I'm really, as I said, this is really quite an open question. I think I might just jump through these videos because they're a little bit long, but this is, for example, an ant that seems to be taking a little bit longer and repositions the object several times. This is a hugely slowed down video, so I think it's going to take a while to see. Let me see if I can skip along a little. But you can see it's kind of pushing it between its legs, deciding whether this is a good grip. And then eventually, once it's happy, it will head off. But it will sometimes put it down again and pick it up again to get the better grip. So it's not just some kind of completely blind, close my mandibles and see what happens. There seems to be some kind of decision making going on. How do they decide whether they're going to try and lift an object and run with it or drag it So we actually started this study because we're trying to make the ants go backwards to make their navigation difficult, because how do you recognize the visual route if you're going backwards along it? So this is how we came up with these kinds of hollowed out cookies, but it turns out the ants are pretty good at working out where to grasp this, and that it's not worth trying to pick it up, but that instead they should try and drag it back home to the nest. And this, again, it's an individual ant will be doing this kind of behavior. And then also they will often appear to make some selection. They don't necessarily just pick up the first thing that they run into. So in this case, this ant seems to take a little while to search through these different crumbs to decide, and we don't know whether it's deciding, one of them is just a fresher, better smelling crumb, or it's actually trying to say, well, what's a good size for me to take back, because it should be more efficient if they take back the biggest thing that they can carry at a reasonable speed. So here, it seems to decide, in the end, I think it decides against this bigger item, but, oh, actually, maybe it does take the big one. Yeah, I can't remember. Yeah. Okay. And then finally, this is another little cruel experiment we do on the ant. So this ant is dragging this thing backwards, and we make it difficult so that it actually gets disturbed and runs away. Also, when they're going backwards, they sometimes stop and let go to look around to try and see which way they're going. And while it's away, we switch things over. And the ant comes back, pretty much to where it left the item, but it seems to be a little bit worried that this nice big bit of cookie is gone. But eventually it just compromises with this tiny piece of cookie. And off it goes. Okay. So as I said, this is very much work in progress, and I'm very happy to talk with people about this further in the next few days. And then finally, I just want to mention the most recent project, which is the project that we're actually collaborating with Festo, is a question of, is insect vision actually low resolution? So insects have, ommatidia, each of which is like a single lens with some receptors underneath, and it's long been thought that limits their visual acuity. Basically, the size, the input for one lens should be the equivalent to 1 pixel for them, and that's somewhere between 2 and 4 degrees of visual angle, which is quite low resolution. However, my new collaborator in Sheffield University, Mikko Jussila, has been doing some amazing experiments where they've found, so not only, it's been known for a little while that the whole retina of the insect, actually there's muscles attached that can pull the retina. So even though you can't see anything moving on the outside, there's something moving on the inside. But he's even found that individual photoreceptors also make little movements. So every time they get hit by enough light, they actually do a movement, which means they sweep their receptive field back and forth. And so far in theoretical models, and now we're going to try and show it in lots more modeling and trying to build some systems as well, this has the potential to actually increase resolution by at least 4 times. And moreover, could the actual pattern of movements, which is this kind of, they do this mirror image movements in the 2 eyes, might actually allow the insect to have stereo vision. So we always thought their eyes are too close together for them to have stereo vision. But maybe at least for some interesting operating range, for example, the range in which they're doing manipulation, they may actually have stereo vision as well. So that's our kind of really up to the minute coming next project. Okay, so I'll finish there. I'll just say thank you to all the people in my lab, the previous people, the current people in my lab, my collaborators, and people who have joined me as partners and as funders. Thank you.